Hi everyone, welcome to the next video. We start our discussion of the first evolution experiment that we want to discuss. This is a study that that was published in 2016 in the journal Science from the group of Roy Kishney at Harvard. So this study asks that how easy is it for evolution of antibiotic resistance in bacteria. So the first, historically we discussed this briefly in an earlier video, that antibiotics were discovered in the first half of 20th century, and when that happened it was thought that we found this great tool to control the growth of bacteria in places where we didn't want them growing. So suppose we had an infection, you take antibiotics and the bacteria are taken care of. However, in the decades of the second half of 20th century, we soon found out that bacteria do not just statically stay while antibiotics are being used. They can change their physiology via accumulation of genetic changes, so that they become resistant to antibiotics, and an antibiotics that was working historically would no longer be functional in the case where antibiotic resistance has been acquired. So that is why antibiotic resistance is such a big health concern today, because because of the overuse of antibiotics, there are so many pathogenic strains which are resistant to many antibiotics that we know. In some cases we have isolated strains where hardly any antibiotic that we know works, because the bacteria is resistant to everything that we know. So when we think of antibiotic resistance, before we discuss the results of this paper, it's a very nice 2-minute video that the authors produced, which we will look at in just a few minutes. But first is that if we have a bacteria, when we say this is resistant to an antibiotic, so what that means is that this can resist clinical level of antibiotic. So let us say this bacteria is not resistant. So we say that this is a bacteria which is susceptible to an antibiotic. What that means that, so let us say the antibiotic we are talking is called trimethoprim. That is just one of the antibiotics that is commonly studied, but it could be ampicillin, it could be tetracycline, any antibiotic you like. So this is susceptible to trimethoprim. What that means is that there is a concentration of antibiotic. So this is the bacteria, that this bacteria will grow in an environment if antibiotic concentration is less than C naught. If the environment has concentration which is greater than C naught, then this bacteria will not grow. This concentration below which growth happens and above which growth does not happen is called the minimum inhibitory concentration of an antibiotic. Minimum inhibitory concentration of an antibiotic against a particular strain. So let us imagine that this is a scale which indicates antibiotic concentration. This is 0, this is very high, and in between we have intermediate ranges of intermediate values of concentration of the antibiotic. Then because this strain is susceptible, its MIC would be towards the lower spectrum would be something like this. So, this strain has an MIC which is here. Let us call this C naught. So, it can exhibit growth if grown in an environment which contains antibiotic in this window, but this cannot grow if it is grown in an environment which contains antibiotic in this window. So, very small MIC. However, what can happen is that this individual can evolve to acquire mutations. This is the genome, and it can acquire mutations, and these mutations can facilitate it becoming resistant to the antibiotic. And as a result of this, the MIC associated with this new genotype will increase and maybe go here. This is the new MIC. And maybe if we keep increasing the pressure on the bacteria by using more and more antibiotic, maybe we are facilitating selection of newer genotypes which are even more resistant, and by that what we mean is whose MIC is even higher than the strain that we just described. Maybe these sort of changes are getting selected for, and as a result this moves to further higher MICs. This is the MIC of the third strain, and so on and so forth. So, this MIC values can change depending on what is the selection pressure that we are applying. So, now let us think of an experiment where we are growing these three strains in an environment. Let us imagine an experiment where we are growing these three strains in an environment where the antibiotic concentration is given by this value. Let us call it C1. If growth is taking place in C1 and all three of these genotypes are present, let us call them A, B and C, then what is going to happen is that because C1 is above the MIC of B, is also above the MIC value of A and B cannot grow in that environment. Only C can grow. So, in this environment at C1 concentration, strain C is going to win. This is going to get selected, the other two cannot grow, and hence they are going to go extinct. However, what happens if we do this experiment at this concentration? Let us say C2. Since C2 is above the MIC of A will go extinct, but B and C should both be able to grow in this environment, and that is indeed what will happen. But what typically happens is that increasing MIC comes at a cost. Because you are investing cellular resources towards becoming resistant, the growth rate of these two will not be equal, and typically what will happen is this individual will have a higher growth rate as compared to C. So, in this environment B and C will both grow, however B will likely grow faster. As a result of this, selection will select B, and eventually if long enough time is given, B is going to get selected and C is going to go extinct and A will never grow. So, this phenomena is the cost of becoming resistant, that you became highly resistant, and if the environment has a lot of antibiotic, that resistance is going to come in very handy, because in this case only C is going to survive. However, if the antibiotic in the environment is lowered to C2, then A is going to go extinct, but between the competition of B and C, B is going to win, because C, in order to become highly resistant, paid a cost in terms of its growth rate. And by the same token, if antibiotic concentration was here, perhaps A will survive, and so on and so forth. So, all these dynamics are there. There are trade-offs involved, that the more resistant you are, the more slower your growth rate in the absence of antibiotics. So, again fitness is a very environment context dependent phenomena. In an environment where this C1 is there, C is the fittest, but in an environment where there is no antibiotic, A is going to be the fittest. So, environment decides which genotype is the fittest in a context. So, suppose in this experiment that we are going to discuss, suppose we want to study how can we evolve E. coli, how easy is it? The question we are asking is, how easy is it for bacteria to become resistant? And becoming resistant in this context means just increasing MIC. So, how would we design such an experiment? So, in this study they use a very clever approach to answer this question. What the authors do is they take a plate which is not liquid, because if it was liquid then everything would mix with each other. This is not solid, because if the plate is solid then bacteria can't move in that media. This is a semi-solid, this is typically used for studies where we are studying the ability of bacteria to swim. So, this is a plate which is very large in dimension, its dimensions are of the order of several feet by several feet. Typical Petri plate is only about this much, this is I think 4 feet by 2 feet, and they divide this plate into several sections. The media in this plate is so that bacteria can swim. And that is important and it will become clear in just a second. So, they divide this plate into several sections and this, at the start of the experiment they introduce bacteria at the left edge of this plate as I am showing. So, I just want to give you a context before we watch this 2-minute video which exactly will show us what happened in the experiment. So, at this edge they introduce bacteria. Now, what is going to happen is that these bacteria will grow, they will divide and they will consume the resources that are available near this edge. As they will consume resources, all the resources near the edge are going to get used up, and as a result bacteria will sense this gradient that all the resources here are gone, but the resources a little bit away are still present. Bacteria have a sensory system which is called chemotaxis system, through which they can sense gradients and move up a gradient if it is attractant that they are looking at. So, as a result bacteria will move up the gradient and they will start consuming, and very soon we will see that bacteria start spreading in this direction. As they are eating resources, they are swimming and moving towards right. Now, these dotted regions indicate split in different parts of the plate, and the first region has no antibiotic. So, in this region there is absolutely no problem, the bacteria keep eating and keep moving to the right until they hit this first barrier, beyond which in this region of the plate there is antibiotic at concentration equal to the MIC, and this is the ancestral strain. So, it would be worthwhile if you just pause the we'll just take a 10-15 second break here, and I want you to think about what would happen as these bacteria are moving towards the right and they hit this frontier beyond which there is MIC concentration of an antibiotic. What would happen as you see this bacteria moving right, hitting this frontier, what would happen thereafter? I will just give you 15 seconds to think about this problem. So, what you should note here is that once bacteria will reach here just fine, but once they are here at this front, they can't grow beyond this because this region has MIC concentration of antibiotic. They can't grow. As a result of that, they will pause here. And now growth will not happen until an individual here acquires a mutation which allows for growth in MIC concentration of antibiotic, and this individual will now spread, and this spread will continue till the bacteria reach this front, at which point growth will halt again because this region has concentration of antibiotic which is 10 times MIC. And the same phenomena is going to keep on repeating with every rightward section of this plate has 10 times the concentration which was present on the section to its left, and so on and so forth. So, the question that they are asking is that how far can bacteria go towards acquisition of this resistance. And when this paper came out in 2016, the authors of the study also made, when this experiment was going on, it was several days long, they made a video of the entire process and they added a dye to the plate background so that you can, the visualization of bacteria moving on the plate is made easier. So, we can actually see this experiment. So, they condensed something like 10 days to 2 weeks of experiment into a 2-minute video that is present on YouTube, which is what we are going to watch now. And we can actually see what happened in this experiment as concentrations go up. So, let us watch this 2-minute video and we will continue our discussion after that.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short.
Okay, so that was the hopefully you got the gist of it. So we'll walk through what happened here. This is a petri plate which is 4 feet by 2 feet in dimensions, and the challenge is sort of maintaining this soft agar and allowing for growth and keeping the whole condition sterile for a period of 10 to 11 days. So they made this lovely video out of this, and as they highlight in the video that this plate is sectioned into these concentrations. So 0 indicates no antibiotic, and then one is 1 times MIC, and then 10 times MIC and 1000 times MIC. So, what you are looking at in this video is evolution of resistance to an antibiotic, but 1000-fold. A bacteria that couldn't live at 1 times MIC is able to survive at 1000 times MIC. Now, is that a very hard thing to ask of a bacteria, or is that a very easy challenge to ask of a bacteria? And before this, we didn't really have a good sense of it. And then of course, they introduce the left and the right halves are just mirror images of each other. So, they introduce these bacteria here, and they quickly occupy, both from the left and the right, they quickly occupy the first band where there is no antibiotic, and that is not a challenge. They are just using up resources and spreading and filling the space available. But then interesting things happen, that now once they arrive at the interface between 0 antibiotic and 1 times MIC, there is a pause, because this genotype that expanded in the 0 region cannot occupy the 1 region, because it can't grow and survive in that antibiotic concentration. So, there is a waiting time associated here at this front, that one of these individuals should be able to pick up a beneficial mutation and then grow in the subsequent section of the plate. And then we begin to see this happen, that here is where the mutation first happens, and it starts to spread radially. These are the progeny of this mutant that arrived at this particular location on the plate. However, as you can see on the left section, something interesting is going on, that both on the left and the right actually, that mutation that has facilitated growth in this next section has not just happened at one place, but there is 1, 2, 3, 4, 5, 6, 7, at least 7 places where this mutation has happened, and all 7 are now spreading into this region. In fact, we also see more mutations coming up on the right hand side where these mutants are now spreading. So, they quickly grow up and then fill up the space, and then again pause at the next front, and not everyone that was able to grow at 1 times MIC can grow at 10 times MIC. As you can see here, that there is a mutation here which quickly grows to expand in the 10 times MIC region. This mutation is showing an interesting trajectory of growing in 1 and then 10, and so on and so forth. This growth process continues, and every time it hits a section of the plate, there is a pause associated with acquisition of new mutations. And eventually this plate is occupied by the bacteria evolve, they acquire mutations to be able to grow in a region of plate which has 1000 times its MIC. And because this is a plate, I can sample bacteria from different parts of the plate and actually study where mutations were acquired, which mutations were acquired, and so on and so forth, and completely understand the evolutionary trajectory of these populations as they evolved from their ancestor state to a state where they grew up, they acquired 1000 times its MIC. It is a fascinating experiment, and interestingly they collect bacteria from different parts of the plate, and this is the study, this is the lead author, and what they find then is that from the final map that we saw of the entire plate, we can isolate bacteria from different sections of the plate. We know how they performed on the plate because we made the video. We know which one came early, which one spread faster. So, we know their relative fitness. We also know which part of the plate. So, we know that which antibiotic conferred how much resistance, and that is linked with, and then we can isolate these bacteria from the plate and get their genome sequenced and identify that which regions of the genome were targeted the most. So, for instance, in this one, x axis represents the nature of mutations that happened, that which genes were targeted, which genes acquired mutations, and y axis represents that how many times was that particular gene targeted in the entire sampling that I did from different places on the plate. So, as you can see that there is a gene called FOL-A which encodes for a protein which is responsible for folate biosynthesis and is also associated with resistance to trimethoprim, and there is a lot of study on this gene being done. That was targeted almost 50 independent times on this plate. Then we have genes something like MAR-R, which stands for multiple antibiotic resistance. Then we have another gene called SOX-R, which is associated with superoxide, which is associated with handling superoxide stress, and so on and so forth. And then the frequency quickly decreases a lot. Some of these occur only 1 or 2 times, and there are many mutations which occur only once on the entire plate, no matter where you sample it from. But there are these signatures of conserved evolution that if you evolve bacteria in a particular stress, certain regions acquire mutations and so on and so forth. So, we can look up the reference, but the idea is that we can do this sort of an experiment and we can trace the phenotype with the underlying genetic changes that are taking place in the organism. Interestingly, one last thing before we wrap this experiment up is that the authors did the following experiment too, that they divided the plate into 2 regions. Left region had 0 antibiotic, while the right region had 1000 times MIC. Remember, in the original scheme that they have, the regions are 0, 1, 10, 100 and then 1000. So, you allow for a more graded response in the actual experiment, but they also did this experiment where you only have binary regions in the plate, either there is no antibiotic or there is 1000 times antibiotic. And when you do this experiment, obviously the bacteria quickly spread through the left half and occupy this region, but then they come and halt here just as they do in the previous one. And once they are here, now you are waiting for a mutation to take place at this front, such that because of that mutation MIC goes up 1000 times, and that mutation never happens. So, what that means is that the challenge that was posed to the population here in going from 0 to 1000 was too steep a challenge, such that no one mutation could accomplish it. This never happened. Whereas, when you do it in a more graded response, you saw that antibiotic resistance could be increased by a factor of 1000 in a matter of just 10-11 days. So, that was the first experiment we discussed. Next, we will move to a classical E. coli experiment which has been going on for more than 30 years now. It is called the long term evolution experiment that we will start with in the next video.